the knowledge that humans have gained through their study of genes and genetics has led them to the technology called genetic engineering, which is the ability to deliberately modify the characteristics of an organism. One of the greatest advantages, the Human Genome Project, abbreviated HGP, helped advance our knowledge of genetic engineering. This was an international project done by scientists all over the world to actually map out the genes within the human genome. Through the Human Genome Project, we now know exactly which gene is found on which chromosome and what the sequence of the bases is within that gene. The technology used to work out the human genome can also be used to work out the genome of viruses. So if there's a new disease that breaks out, you can map the genome of the virus so you can improve treatment. You can also know exactly which mutations are linked to which type of cancer, and that could lead to better diagnosis of diseases, early detection of diseases, and even better treatment as well. An example of this, we can see the two genes BRCA1 and BRCA2 that are found on chromosome 17 and chromosome 13 respectively. These two chromosomes, if you have both of these, you have a very high percentage chance of developing breast cancer. Now, you might know this or not, but something interesting is that Angelina Jolie, a few years ago, because her mother died of cancer at a young age, she had herself tested and found out that she actually carries both these genes, and she decided to have an elective surgery, a double mastectomy, to actually remove her breast tissue so that she does not get breast cancer. Another advancement in the field of forensic science is that through the Human Genome Project, we can take very small samples of blood and actually say exactly what the individual looks like that this blood comes from. So you can actually collect very small samples of blood from a crime scene, and if you test it, and through DNA fingerprinting you see it's not the victims, you will guess obviously that it's the suspects, and testing that and genotyping that, you can actually say, oh well, this person that did the attack, the suspect, is of a certain race, with a certain skin color and of a certain descent, like Spanish or something, so the police can look more accurately for a suspect. With this technology, we can also modify and change various crops, animals, livestock, and even insects. So all of these we'll do in a little bit more detail a little bit later in this video. Some bacteria have the ability to digest toxic waste and industrial waste, and if these bacteria are genetically engineered, they can actually work faster and do more work and more breakdown of other harmful substances, so it's useful for the environment. Human DNA can also be used to trace ancestry. So your genes will say how much European you are, or how much American you are, or how much African you are. And in that way, human migration patterns in the past can be followed. But we will do this in more detail when we look at human evolution. So if these are the possibilities, what is actually being done in the field of genetic engineering? And we're only going to do a few examples that are important for the exams. With the discovery of the enzymes called restriction enzyme and ligase enzyme, scientists are actually able to cut DNA at certain sections and insert desired genes and join them again to form recombinant DNA. Now this is put into practice when bacterial DNA called plasmids are used in the medical applications to produce artificial insulin. Now this is human insulin. The bacterial plasmid is removed from the bacterium cell, and the orange little circle you see at step 2 is cut open, that's the DNA, is cut open with restriction enzymes. Once that's cut open, a shorter gene shown at step 3 in red And a gene that makes insulin is cut out from a healthy person's cell and then inserted into the bacterial DNA. And then at step 4, you see how this new recombinant DNA, which is partly human and partly bacteria, is put back into the bacterial cell. The bacterial cell then starts to produce insulin, which is used to treat diabetes in humans. Gene therapy is also an attempt to actually treat diabetes. What happens here is that scientists will genetically engineer a virus by removing its own genetic material and inserting the gene from humans that make insulin into the virus. The person is then infected with that virus to deliver the desired gene into the pancreas of the person. Cloning is another way of genetic engineering. Now, we actually have 2 types of cloning: therapeutic and reproductive. The one that you probably know best is reproductive cloning. Reproductive cloning is the procedure that was used to create Dolly already back in 1996. She was an identical genetic copy of another little sheep. The problem with this is that Dolly was the 277th attempt. There were 276 failures before they succeeded in cloning her. This is one of the reasons why reproductive cloning in humans is not a very popular idea. Most countries, it's illegal, because what do we do ethically if we fail 277 times in human trials? To clone an organism, you actually need a somatic cell, which you're going to remove the nucleus of. Then you're going to have an ovum, of which you remove the nucleus as well. You will then insert the nucleus from the somatic cell into the ovum and give it a little electric shock, and it starts to develop into a whole new organism. The cloned embryo obviously needs to be planted into a surrogate sheep so that it can grow during a normal pregnancy. In order to move the nucleus from one cell into another, a technique that is called somatic cell nuclear transfer is used. This micrograph shows you at number 1 how the ovum is held in position and a very small needle is inserted into it to remove the nucleus. So remember, an ovum is in actual fact the size of a full stop on a page. So just imagine how small the needle is that they insert in there. The problem is that reproductive cloning is going to produce a whole new individual. But what's the point? I mean, there are so many amazing people on the planet. We don't want to make a copy of them all. So ideally, what scientists are now trying to do is to use therapeutic cloning. So instead of cloning a whole organism, you just want to clone parts of it. In other words, you want to clone some stem cells. You want to get a part of that person copied. Now, we actually have different types of stem cells. A stem cell is a type of cell that hasn't decided what it's going to be yet. It's not differentiated yet. Originally, the main type of stem cell that scientists worked on was called embryonic stem cells. But that's an ethical problem because they got those from real living human embryos. Then they started working on tissue-specific stem cells that they got from an organ, which they could grow in a lab into the organ which it came from. But that was also a problem and not always very successful. So more recently, they've started working on induced pluripotent stem cells, which means they're able to take a tissue-specific stem cell from your body and cause it or convert it into a stem cell that will act like an embryonic stem cell. And from that single cell, they can clone many of those cells and produce only an organ for the person that needs it. The very latest discovery is CRISPR-Cas9. Now, that is a way of permanently editing genes really fast. So scientists can cut add, remove any gene they want very accurately, cheaply, and very efficiently. But the problem with genetic engineering is that it's very unethical sometimes. The question arises like, which genes are we going to change? And for what gain are we going to do it? And all of these issues will be looked at in more detail in the next lesson.